Hello everyone. I will be reporting about the Hippurate test or the Hippurate hydrolysis test. So this test is one of the many different biochemical tests that can be used to distinguish different types of bacteria based on their metabolic capabilities. So this report covers its purpose, principle, the reagents used in the laboratory, the procedure, the interpretation of results, and its clinical significance. The purpose of Hippurate test is to determine the ability of the organism to produce hippuricase, which hydrolyzes the substrate hippurate. It is primarily used in the identification of Campylobacter jejuni, Listeria monocytogenes, and Streptococcus agalactiae. So the Hippurate test is based on the ability of the organism to hydrolyze sodium hippurate to benzoic acid and glycine by the action of enzyme hippuricase. So particularly, hippurate is able to hydrolyze, that means by using water to break bonds. So hippurate is able to be broken down by water if an enzyme called hippuricase is present. It uses water to break down hippurate to make the products benzoic acid and glycine. And Hippurate test involves ninhydrin as the indicator, which reacts with any protein or amino acids, and in this case, detects glycine. So in the Hippurate test, glycine is the product that is detected by an oxidizing agent, and that is our ninhydrin reagent. So if glycine is present because Hippurate has been broken down, then this will give us a deep blue or purple colored complex. So the reagents used in the laboratory, we have Hippurate solution. Hippurate solution can be found either commercially in the form of dehydrated tubes or as discs or tablets. Pero pwede rin siyang i-prepare sa laboratory as a 1% Hippurate solution. So for this 1 gram of sodium hippurate, it is added to 100 ml of distilled water. We also have ninhydrin. So this reagent can also be found commercially and can be prepared in the laboratory. So for the preparation of the ninhydrin in the laboratory, 50 ml of acetone and 50 ml of 1-butanol are added to a dark glass bottle. So to the bottle, 3.5 grams of ninhydrin is added and mixed well. So the procedure of the test, first, we add 0.4 ml of the hippurate reagent to a test tube. And after adding the hippurate reagent, a heavy suspension is prepared from an 18 to 24-hour culture. And third, the colony should be picked up carefully as to not pick up agar, which contains protein. And fourth, the tube is then incubated for 2 hours at 35 degrees centigrade to 37 degrees centigrade. So after this incubation period, 2 drops of the ninhydrin solution are added to the hippurate reagent organism mixture. And lastly, the tubes are then re-incubated at 35 degrees Celsius for 30 minutes. So the observation of the tube is made at 10-minute intervals for the appearance of the deep blue color. And the color change will usually appear within 10 to 15 minutes after the ninhydrin indicator solution has been added. Results and interpretation. A positive hippurate hydrolysis reaction is indicated by the appearance of a deep blue color within 30 minutes, and a negative reaction is indicated by the appearance of a faint purple color or there is no color change at all. So for the clinical significance of hippurate test, it is most frequently used in the differentiation of Streptococcus agalactiae, Campylobacter jejuni, and Listeria monocytogenes. It is also used in the differentiation of beta-hemolytic Streptococcus agalactiae from other beta-hemolytic streptococci, and it also aids in the separation of Campylobacter jejuni and Campylobacter coli strains. So that's all. Have a nice day, everyone. Thank you.